Welcome, and thank you so much for joining us tonight from across the country for healthcare, biological research, and AI. We have a stellar cast of innovators for you tonight. I'd like to start, though, by thanking the Yale Club of San Francisco and its president, Rahul Prasad, who is with us tonight, tuning in. I'd also like to thank the Yale Club of Silicon Valley for joining in presenting this. And of course, our sponsors, Yale Women, whose chair is with us and providing this virtual program platform for us tonight, and then the Yale Alumni Nonprofit Alliance. Together, we've been able to bring this to you. Without further ado, I'm Eleanor Pepples. I'm chair of the Yale Alumni Nonprofit Alliance here in the Bay Area, and also very proud to be a board member with the Yale Club of San Francisco. I wanted to sort of launch by just sharing perhaps what you already know, that AI's potential to advance human health is virtually limitless. Tonight, we'll explore the many ways AI accelerates and elevates the quality of biological research and speeds the translation of discovery into cures, and really reshaping biotech and just changing the landscapes for healthcare in so many domains of it. And tonight, we'll focus on AI and imaging in its relationship to improving healthcare and personalizing treatment and interdisciplinary connectivity, which I think we'll find is a theme. So I could walk through our panelists' bios, which are extensive. So instead, what I'd like to do is ask a question that not only fascinates me, but I think will the audience too. So what has drawn each of you to this work, and where are you seeing the greatest benefit of AI in healthcare, bioscience, and from sort of the research as well as in biotech too? Who would like to take that first? Smita, I'm looking at you, but you can't tell.
Okay, thank you. So I guess there was like a two-part question there. First, what drew me to this work? Yeah, I first became interested in utilizing computing in general and AI more specifically. Back when I had just graduated from my PhD, I was working at IBM Research, which is a great place to work, but like day in and day out, I was really working on something small, designing something automatically that went into a computer chip, and it was very far removed from health and medicine. And then I started thinking the same kind of computing that I'm doing could have a lot broader reach and impact if it could impact something like discovery in the sciences, or it could directly have an impact on sort of human health. And so I started wanting to aim my computational skills towards biological discovery. And then back when I kind of switched to that domain back in 2010, I feel like the trend was to then just go and become a biologist. But I'll say I didn't do that. I'm still a computer scientist because I really feel like my skills in developing AI for biology is how I felt I could best contribute. and this leads to your second question, it's really because AI can affect everything. It can affect processes in basic research. It can affect processes in pattern detection, causal inference, all the way down to healthcare, which some of the other panelists may touch upon. And so I think that's what drew me to it, and that's what keeps me here.
Excellent. Dr. Krishnaswamy, I will come back to you in a moment. So I think I know who's coming next, but please step onto this stage and introduce yourself. Would you unmute yourself? Nur-Taz Rahman, please, Dr. Rahman.
All right. Thank you. I love this question, really. So what drew me in general to study genetics and cell biology was exactly what I am doing now, which is trying to understand how is it that there are four nitrogenous bases that define who we are and what we look like and predetermine us for obesity or diabetes, or tells us that or makes us live longer or live shorter. And for me personally, grow taller or shorter because I'm just 5 feet. I know you cannot tell from this remote webinar, but so I've always wondered these things, and that's what kind of propelled me to study biology and pursue a PhD in biology at Yale. And I hear absolutely what Dr. Krishnaswamy was saying, Smita was saying, that there was just, even that was 2012, and even then it was kind of like you become a biologist or you go into computer science and you're building the tools, etc. I was in Diane Krause's lab at Yale in the Yale Stem Cell Center, and she studies hematology. And I also started I went in there with the goal of studying how stem cells that live in our bone marrow mature into these cells that gives us these platelets that essentially form a bandage whenever we cut ourselves, a small nick, and the platelets will come and make a natural band-aid and prevent us from bleeding out, right? And that process breaks down for a lot of people, unfortunately. And so that's what I was studying. But it so happened that Diane already had lots of bioinformatic data in her lab. She was already looking at how do these proteins bind to the DNA, and then what happens to the expression of the gene that DNA encodes. And I was like, wait, this is going really into the detail of what I've always wanted to understand, is how does that gene, how does this DNA tell the cell what to do? And so that's how I went into learning bioinformatics. And Diane was, as a mentor, she was super patient as I figured out how to run code, how to work with computing systems. And she gave me this opportunity to learn bioinformatics. And now this field has exploded and is exploding. And with the advent of artificial intelligence, AI, which is what we are here to talk today, it's going to take this whole field into a new dimension. It's really, for the way I see it, is that biology, all the biological data that we have been harvesting, gathering, and storing all this time, it needs a tool like an AI to digest it and give us more insight. And I think that this is a very exciting time to be part of biology and part of bioinformatics.
Wonderful. Thank you. Now, we have with us from UCSF Medical Center, yet another Yale, a well-decorated and very distinguished Yale, I might add, Dr. Sharmila Majumdar. Would
Thanks,
you give us your insights on that question?
Thanks, Eleanor. I graduated from Yale a few years ago, and few being many, in 1987 from the Department of Engineering and what was called Engineering and Applied Sciences, When I began, there was no bioengineering even as a discipline or a department within Yale. But I started working in the area of imaging, which confounded everybody in applied physics as to why was I going to the med school to do engineering and imaging and so on. Oops. Sorry. I thought I turned it off. Sorry, I apologize. At that time, magnetic resonance imaging was just beginning. It is basically beginning as a new discipline, and I was there at the ground. And when we started, a single image itself would take several hours. So it is no surprise that my goal was to do faster imaging, more quantitative imaging, and to get more data out of the large three-dimensional stacks. That's what led me to work in artificial intelligence and imaging. Imaging is an amazing resource. It gives you insight across the scales. It gives you insight at the level of the cell. It gives you insight at the level of the whole human. And everybody has heard about magnetic resonance imaging. They've heard about CAT scans or CTs and computer tomography. X-rays are everywhere today for all sorts of chest pathologies, for musculoskeletal pathologies, etc. Part of imaging actually is also image interpretation. And part of image interpretation can be very varied across. It often requires skilled readers, especially in global areas, which are, rural areas, these people are not available. So as a result of this, I started wondering, I'm doing a lot of quantitative imaging, I'm doing all of these things. What would make it faster? How could we get this data? And that's why when, neural networks and artificial intelligence became a thing, I started looking at what could we do. We could do neural networks several years ago, but the impact of computational resources made it really possible to apply AI tools to large data sets, such as imaging data sets, and integrating not just imaging, but actually using the images to predict function, like how the body moves, skeletal movement, biomechanics, or, for example, what are the functional characteristics of the cardiac cycle. So imaging enables us to combine, artificial intelligence enables us to combine all these factors. And that's what has led me and attracted me towards using artificial intelligence in AI, in imaging, and in healthcare. And that, I think, is going to be one of the biggest advantages we will have once we can deploy this tool safely and effectively in healthcare.
Thank you. Thank you so much. That's, it's so impressive that you were on the ground floor, really, in the beginning. Are just such an innovator. I admire that. In fact, each of you on the panel. So, Smita, well, you didn't mention that you are an associate professor at Yale with your own lab there, and I wondered if you might tell us a bit about Yale and your arena. What are some of the most exciting things that are happening there? And how does Yale support you in what you do?
So yeah, Yale has been, extremely supportive in what I do. I can share a little bit about what my lab does if it's a good time to do that.
Sure.
I have a couple of slides, and I really feel like Yale is one of the few places where I could do this, so you can see some of my affiliations. So I'm a primary faculty member in computer science and genetics. One of them is in the School of Engineering and Applied Science, like where Sharmila was. One of them is in the medical school. There's a number of institutes where I can collaborate with domain experts to apply AI pervasively and develop new methods of AI for this. So basically speaking, my lab tries to develop AI methods for science that are mathematically well-informed. What do we mean by that? We try to incorporate constraints from the underlying system biology, topology, and dynamics, which are very important in modeling any living system, into deep neural network frameworks. And then we use these models to infer dynamics, causal structure, mechanisms, interactions, and other things from cellular, molecular, imaging data, neural patient data. And I have a number of students very excited to do this on all different scales. I say the difference between general machine learning, which is basically predicting something static about a high dimensional input, and something in the sciences. In the sciences, you really want to learn the rules of the system. And people have been doing this in science forever with modeling so that you can predict out the dynamics of the system. But now we can finally combine the two because we have enough power with the AI so that AI can learn models like virtual models of cells. And you can predict and simulate systems in a way that you didn't used to be able to do unless you had carefully curated a model. Along these lines, we have systems developed for inferring dynamic cellular trajectories from static snapshot data, inferring underlying causal networks, and reasoning about their perturbations. We're also working a lot on molecular data, where we try to represent the geometry, topology, and other parts of a molecule in such a way that we can optimize it automatically using a neural network. And we have some focus on also modeling the context of a neural network. So we have large multimodal foundation models for molecules that model things like sequence structure, biochemical properties, cellular background gene expression, and things like that to predict immunogenicity, splicing, other things. A trend that I'm really excited about that's just coming up in my lab, and we're working closely together with a lot of other labs, including the Gerstein lab, Fabian Theis lab, and other labs, is this idea where we can have a virtual agent that can actually spit out a virtual cell model. So this is kind of culminating in an agent that is so intelligent that it can create a perturbable model and ask it questions. So these are some things that are coming up. And finally, I have to mention that Yale supports innovation in brain sciences and human cognitive sciences a lot through the WTI Institute. And I have a number of internal and external grants to decode brain signals to help people with autism and nonverbal conditions like Angelman's, as well as to model brain dynamics and their perturbations, much like other people are modeling cell dynamics. So that's just a little bit about what my lab is doing. And of course, as you can see, it's highly multidisciplinary. I work with molecular biologists, cellular biologists, neuroscientists, and that can really be only done at a place like Yale, where all these people come together, both physically and kind of in the spirit to work together.
Wow. Now, would you tell me WTI you mentioned?
Yeah.
What does that stand for?
That stands for the Wu Tsai Institute, and actually Stanford has one of them too. I believe ours is a little bit newer, and it was endowed by Joseph Tsai, Yale alumni, and his wife Clara Wu, and it's a center whose mission is to enhance human cognition, basically, but they have a center for neurocomputation and machine intelligence. And when my office moved into their center, I sat next to a lot of started sitting next to a lot of brilliant computational neuroscientists, behavioral neuroscientists, and I started realizing I could use some of these same basic principles of dynamics modeling, interaction modeling, learning interactive causal structure from data to model brain activity and impact another realm.
Thank you. I wanted a little pitch for the WTI Institute because Joseph Tsai was a classmate of mine at Yale.
Oh,
So
okay, yeah.
we'll give him
Great.
a little
He still
PR
comes
for
back.
that.
I think he lectures in some of the law school classes, and I've met him.
Yes.
He's great.
He's also Yale Law 90, I think.
Okay.
Fantastic. So would you tell us a little bit about some of the awards that you recently won an innovation award this year, and then also your lab? Could you tell us about that so we can really understand sort of the practical implications of the work you're doing?
I think the award that you're referring to is the Yale Health Innovation Championship Award.
Yes.
That was
Yeah.
one together with Dr. Ramesh Batra from the Yale Transplant Unit. And just sort of how we met a little bit is very indicative of the stuff that happens at Yale, because he had sent out a mass email to computer science saying, does anyone want to work on predicting transplant viability of organs? And I immediately said yes, mainly because my mom is suffers from kidney disease. And he came to my office actually, and he started talking to me, and then I started talking about my mom, and he's like, what's she gonna do? And I'm like, I don't know. She might go into dialysis. And he just stopped what he was talking about. He's like, no, she can't go into dialysis. You've got to get her on the transplant list. And eventually we did. He did motivate me to get her into the transplant center. She had a very successful transplant. I was a donor, not directly to her, so that was a problem very close to my heart. And what we ended up working on was taking data from Yale New Haven Hospital and also other affiliated Yale hospitals at the time. And we got the training data to look at people who were on life support, their continuous monitoring, their electronic health record statistics. And we basically predicted out if the person would pass within a time frame when their organ could be transplanted, which in the case of liver is something like 30 minutes. I could be getting that exact time frame wrong. In the case of kidney, it's a little longer. I believe it's 120 minutes. And our model was again a dynamic model. It was what's called a neural ODE with recurrence, a recurrent neural ODE, and this model had accuracy of over 90%. And now we're hoping to take this data and collect data from the larger UNOS database. And I've gotten additional support to try to do that, so we have to get on that.
Goodness. What an excellent illustration of how AI can improve, and I should say, the genius of the individuals guiding it, of course. Thank you, Smita, for that.
Also, my students are Ramesh. Yeah.
And your students, yes. In fact, now that you brought up students, the other day when we were preparing for this program, you mentioned some students who founded both Ascent and another company, Latent. Would you just speak very briefly to how your lab spins off some of those companies?
Yeah, sure. I think my lab, really, the role my lab has in it is just that I believe we have a very innovative, very collaborative atmosphere where all my lab members are friends with each other. So they often say they don't treat each other like coworkers. They're actually friends. They'll go later and hang out at home and, play video games and talk some more. So it's a very collegial, I'd like to think very, very nurturing place, but it's really, once you set that environment going, it goes quite far, because the students who come to Yale, as you guys all know, are brilliant. And so they initiate a lot of it themselves. So Egbert, together with an MBA student, and I believe he was initially paired up during the Nucleate Bio Incubator program that was happening at Yale, the Yale chapter of that. They came up with the idea of Ascent Bio, and the idea of Ascent Bio is actually something that I initial idea of Ascent Bio is something that I actually sort of showed on the screen here. And that's the idea that if you represent your space of molecules in such a way they're not only organized by similarity in structure and sequence, but also by their biochemical properties, then you can actually optimize that molecule by going up a gradient of a property, and you can make a molecule fitter for a particular property and then decode it out. So that's what Ascent Bio actually means, that they're trying to improve molecules. Now they've transitioned a little bit to becoming a molecularly intelligent agent. So it's an agent you can ask human questions about molecules and how to improve them and things like that. So that's an exciting one. A newer one is was started by Yale students who were also EIR, entrepreneurs in residence. So now Yale has some older students who are already executives at companies who are doing sort of an MBA in entrepreneurship, and some of them join Yale Ventures as an entrepreneur in residence. And one of our entrepreneurs, David Kolb, together with another Yaley, David Lewin, started this company called Latent Alpha. And that has a lot to do with using the geometric intelligence that we develop in our lab via a lot of our tools like Fate. And it's they're taking like a broad sweep of improving processes in many industries, healthcare, finance, etc.
Thank you. You know, it was interesting something that you were saying, and it reminded me of an initiative that our new president of Yale, Maury McGinnis, one of the many different contributions that she has made early on when joining our community, which is the Cultivating Conversation Initiative, aiming to foster a mindset of openness and curiosity. And what you're talking about really illustrates that that coming together from different disciplines, with a real desire for dialogue and engagement. I appreciate that initiative is really to work through difficult conversations, but the concept of cultivating conversation to really stimulate creativity is really something powerful that that you were showcasing there. So really another question comes to mind, and this will segue into some of the other panelists as well. But could you tell us a little bit, Smita, how you see, AI, and you've touched on it, improving the quality of and accelerating biological research in academia and industry. And then I'd really like to start to think about some of the beneficial deployments and then transition, you know, to how it can really leverage the field of biology more efficiently and with more confidence in research.
I mean, so I do think AI can help. Biology is sort of like a vast field, and people have different specializations and there's different silos of data that are created. And I think AI can really help systematize the understanding of biology, the knowledge, the way people mine data for patterns, causal associations. It can leverage a lot of the data that's being generated to create sort of unified cellular models that can then be sort of simulated, these kind of virtual cells. But the other part of your question, which is interesting, is what can we do in biology to kind of leverage AI or make it more amenable for AI? And that's a really interesting question, and I think that's starting to happen. I think it basically amounts to breaking down some of these silos. So it's hopefully having biological data in a unified place where it's searchable, downloadable, where machines can access it and train it. Recently, even on bioRxiv, I've seen sort of couple of methods that can actually search through biological data, SC base count, metagraph. I've just seen them in the last couple of days that can search sequencing data. And I think it's really making metadata easy to access. Papers are fairly easy to mine for things like ChatGPT, but the data is often not accessible or the metadata is not correctly available. Making these things available would make it more amenable for AI to work on it. So I think it kind of does go both ways. So biologists can do something about how they store and document their information. Also, another thing that would be really my personal dream data set, because I'm starting to work on biologist, like scientist agents, is if biologists documented if you got a problem, how do you go about designing experiments to tackle it, right? So once we have these data sets for supervised training, we could probably leverage AI to design the next phase of experiment. So that's something that I'd like to see.
Oh, fascinating. Maybe later I'll ask you what happens when they hallucinate. I want to quickly go to Dr. Nur-Taz Rahman. So she's joining us from industry and 10x Genomics, where she's a senior scientist. But she's joining us personally with her insights from industry and is a Yaley as well. So one of my many questions for you is, how are AI tools influencing biotech drug discovery and the virtual cell hypothesis from your perspective?
Tremendously. So let me take that question part by part. So for biotech and the industry, one of the goals is to find therapeutics, is to find those drugs that, drug discovery right now takes some 10, 15 years, a long time. And then a lot of drugs are failing on that path to them getting through the trials, the phase 1, phase 2 trials. So there is a lot of waste that is happening there. So AI is going to accelerate that process. And it is going to do that in one of the things that I really love. And one of the things I'm passionate about, and I wish it was there when I was in graduate school, but that time has passed, right? So what that is the hypothesis generation that we can now do with AI, because it is able to grab all these different data sets that are being generated. So yes, I am a senior scientist at 10x Genomics, and 10x Genomics is one of the pioneers for the single cell technology, which is allowing us to see what is happening in each cell of our body. And it just seems that the timing is perfect. The 10x Genomics started in 2012, and now by 2022, we were already talking about artificial intelligence and how it is going to help us. So one of the things with this hypothesis generation that I really like are some software now, some AI tools that are now out there that can help us talk to these single cell data sets. Okay, so in 2024, by some estimations, there were about 5,000 to 6,000 single cell data sets that were generated in the world. Okay, so all these single cell data, we can train these AI tools to ingest this data and then talk to us. So some of these are cell to text, cell to sentence. There is a chat cell. We can just directly ask them, what are the top 10 genes that are upregulated in these T cells? What are the top 10 genes that are downregulated in these tumor cells that I see over here? And that is and that is really, really changing how we can go for drug discovery, how we can target specific diseases. The other thing with AI is what Smita has mentioned and her lab is working on is the virtual cell, modeling healthy cells and disease cells, and then perturbing them, telling the AI that, okay, if I now add this molecule, if I now add this protein, how does that change things? What is going to happen to the disease state of the cell? And I think these kinds of questions, queries, these kinds of hypothesis testing that now we can do with AI before we go to the wet lab is a huge time saver. It's a huge resource saver in terms of animal testing that you need to do, in terms of all the reagents that you need to spend research dollars on. You can now do more testing on the AI tools before you embark on some long, tedious journey for even just finding a potential therapeutic molecules that can help with solving disease problems. So that's about the virtual cell. And one of the exciting tools that I have seen and I have been able to play with for this has been AlphaFold 3. which has been worked on and created in Google DeepMind. And they actually, the person who created it, Demis, won the Nobel Prize for it in 2024, the Nobel Prize in Chemistry. And what AlphaFold 3 can do is you can give it any DNA sequence and a protein and even ions like calcium and sodium ions that are in our body, and ask it how will all these fold together, and it predicts it, and it shows you a 3D model that you can play with. You can move over and you can find out exactly which part of the gene is in which part of the DNA is interacting with which part of the protein. And this is fantastic. If this tool was available when I was doing my PhD, I would be able to see that, okay, this is what I see from my bioinformatics. What does it actually look like that this DNA is binding to? The protein that I was working on was serum response factor, SRF. And how does that bind to this part? And can I bring in another transcription factor, cofactor, and how would that bind? So this kind of hypothesis testing is going to really accelerate what the industry as a whole kind of does to help cure diseases by finding molecules and therapeutics.
Wow. You see, this is why I call you all the talent. Now, could you give me maybe a couple of examples where it has led to some cures that might not have been possible before AI? Are you feeling it's leading to more sort of high quality results faster? Is it exponential? How might you answer that?
I think we are still a little bit early because these tools are getting built, and it actually touches on another thing that, I think is important for us to remember is the data sets that we are building and curating and putting out there. They have to be really good quality because that's our training data set to train the AI. I myself am not aware of a specific cure that has happened, but I know that in vaccine development, AI is really helping, especially my eyes are on this on this company in Denmark called Evaction, and they use AI immunology, which is their company AI immunology tool. It's their proprietary tool that they use to predict what kind of vaccines are needed for specific people, because their tool can predict based on each person's genome what kind of antigens, what kind of molecules would best work for them.
I can chime in here. Actually, I'm using AlphaFold 3, which you mentioned, and we're using it in our tool called Immunostruct. The purpose of Immunostruct, one of its purposes, is to design cancer vaccines. So you can put in a peptide here, and you can predict if it'll have an immune reaction or not. And now, as it turns out, previous models would just look at the sequence of that peptide, and it could be a mutate. Usually, they'd put in mutated sequences from cancer patients. So they DNA sequence a cancer patient, and they say, here, the patient has a mutation. We'll take some sequence around it, and we'll just create a cancer vaccine to make the cancer fight your immune system. But it turns out that had very low efficiency. So what we're doing with Immunostruct is, in addition to the sequence, we're modeling the structure. And not only are we modeling the structure of the peptide, we're modeling the structure of how the peptide is presented by the MHC. And now we're looking at also the structure of how the TCR binds to it. And AlphaFold 3 can model all those jointly. And so we can just generate that structure. And that improves our performance by quite a bit. You can actually see it in our preprint paper. And we've already commercially licensed this tool. So people are already using Immunostruct to predict immunogenicity prediction.
Well, I'm trying to follow along, and I'm clearly going to need to read even more articles. You're definitely inspiring me, and I imagine a whole slew of people on this, in our audience. So thank you. So, wow, with that education, I'd like to turn to Dr. Majumdar. You were mentioning sort of the whole patient, the whole health of the human being. So how can we bring biology and AI together to improve the lives of patients? Is this already happening? Is it something we can build for? Could you share some of your insights there? You are muted.
Yes, I can. Sorry. Um, I'm just wondering, should I sort of share some slides? Would that be a good way to start?
As you like.
Okay. Let me just put this out. Do you see that?
Yes, thank you.
Okay. Let's just start with that. I'm going to be talking about, continue to share this PowerPoint window. Okay, continue to share. I'm going to be talking about AI in imaging and multimodal AI and how it can affect people. So let's just talk about, in the context of imaging, what does AI really mean? AI can be used for imaging, as I talked about earlier, making it faster, safer, reduce all the artifacts that are often seen in imaging. Say somebody moves and you don't get a good image, you can correct for that. And you can, of course, imaging has a workflow associated with it. Prescribing the imaging, when your image is taken, if any of you had imaging, you know that there is an operator sitting outside prescribing the views and so on. If all of that could be automated, imagine how fast the patient experience would be. Imaging generates a vast amount of data, especially if you're looking at multimodal AI, where you're not looking at different types of MR images. In the context of oncology, you're looking at PET images, CT images, and various other modalities. It can refer to tissue level imaging, really focusing on the tissue. It can also be whole body imaging, integrating all of this, understanding the features in an image, whether it's normal or it's a pathology, can be really important. And getting that right impacts patient care. Using those images to then see how the body is functioning, that is going to affect patient care. Imagine you have pain in your knee, and I'm going to focus on musculoskeletal because that is my area of expertise in interest. Even though I do other body parts, I feel it presents a really great example for everybody. Athletes have knee pain, old elderly people with degeneration and joints have knee pain, and it affects everybody across the board. When you've cured all the diseases in the world, if you don't have a functioning musculoskeletal system and not moving, you're going to be in trouble. You're going to be immobilized, and that is no fun. So, if you can predict with imaging and these non invasive methods how you will be functioning and moving, it will have tremendous impact, not just on a patient, but for generations to come, for longevity. You're trying to increase your length of the amount you survive. Please let it be with a good musculoskeletal system, because you know what a pain in the back or pain in the knee might mean for you. So, having said that, one of our students, for example, Gabby Hoyer at UCSF and UC Berkeley in the bioengineering program, used a vast data sets of images for looking at joint degeneration. Joint degeneration leads to knee replacement. What she did along with a team is basically look at this vast data set, characterize each of the tissues in the knee, include the bone, the meniscus, the cartilage. These are all tissues in the knee, and basically worked out a scheme of digital twin analysis. Imagine this: you are matched for age, gender, body weight, and pain levels. You're matched with the person, but one person gets a knee replacement in time and another person does not. Are there features within the tissues distinguishing those people? And she found certain features. Imagine what this could do. In the next generation, you essentially use those features, you extract those things from these multimodal images, but then you have decision making tools. You know which of those people with what kind of features, joint features, and what other demographics will end up going in for a knee replacement. You know all of those things. You've created these models. You go in to see your doctor, you're sitting there quietly and you're telling him all your history, and all of that is being done with prompts from some kind of an agent, AI agent. All of that is being collected. it's being pushed back and you're being fitted with a model. A model is analyzing where you fall in this entire survival curve for osteoarthritis or joint degeneration. This is what we are looking at. We are utilizing vast, large data sets, and we are basically coming up with ways to treat something. It might seem inane as knee pain, but will that knee pain ultimately result in knee replacement later on in life? So that's how it's helping people. At UCSF, we have sort of a mode, it's called Information Commons, where all the imaging data, all the electronic health record data, the clinical notes can actually be queried. So imagine this vast data set being brought together by AI. We use this in the time of COVID, for example, to predict what the oxygen requirement would be given lung CT images. So we had, in this particular case, we needed more data than was available at UCSF. We actually did a nationwide, an international collaboration where the data was shared using a methodology called federated learning, where data security was preserved. Everybody kept their own data, but the weights were shared. So this kind of large data set integrating large amounts of data has enabled us to also address another important problem. For example, the opioid prescription in chronic lower back pain is one of the leading causes of opioid addiction in the United States. Addressing some of these things by combining imaging, by combining other factors, we've been able to look for what are the primary determinants of opioid prescription in lower back pain. With no surprise, primary insurance does come out as one of the first things that actually makes a difference. So this is, I think, how overall, these are just some examples of how combining imaging and AI and large data sets in healthcare can actually help the patient, starting from making things more comfortable by faster imaging to putting real models in place where when you go in with some features and characteristics such as pain, you can then be put into a model which predicts something down the road. If we are expecting to lead longer lives and exist with disease, cancer-free, heart disease-free, Alzheimer's-free lives, then I think overall an integrative approach, integrating all of these diseases is going to be really important in healthcare. And I think imaging here and artificial intelligence will play a crucial role. And I haven't even addressed integrating the genomics that Nur-Taz talked about, or integrating the single cell analysis that Smita talked about in this entire picture. Everybody gets biopsies, everybody gets tissue cultures. If you could integrate all of this, I think it would be amazing.
Incredible. Thank you so much for sharing that with us.
It's a
Goodness.
little bit aspirational, I will admit. It is, we are not there yet, but I think it's very possible. I think that's what we're all working towards. I think the biggest integration would be if what Smita is doing, what Nur-Taz is doing, what Sharmila is doing could all be integrated. And I think it would make a huge difference to healthcare.
So all of that is so inspiring. And I don't know, Nur, if you and Smita have anything to add there before I go into some of the challenges and sort of ethical questions that come up with the use of AI.
I would just add that I completely agree with Sharmila that we, we need to all pool our resources. And there is also the advent of these models that are called federated learning models, where you don't necessarily have to share the data, but the model is learning from all the data sources. But hopefully there will be, more such agents that are, curating and talking to one another. And the thing that excites me most is initially when these very large models came out that had billion, trillion, whatever parameters, those are large monolithic models. But what I'd like to see is a lot of like small models that talk to each other and share information across barriers. And I'm seeing more of that happen with agentic AI, and that's really exciting me.
Yes, the only thing I want to add, again, like we are standing at the precipice of really expanding on all these data sets that we can now lay our hands on and curate. And one that has become one focus of biology that has become very close.
Nur, we
has
are
been
losing you for a moment there. If you need to turn your video off so we can hear you, that might help for a moment. We all want to hear what you have to say. So I feel like I'm with CNN. So when we have Nur again, we'll come back to you. Can you would you share that with us again? She must be rebooting. So we'll just wait for her for a moment. And again, I just really want to thank the audience and let you know that we have some information in the chat and those questions, and we will promise to get to you after we finish the core of our program. Nur, you're back.
Yes, can you hear me? Because I did
Yes,
not realize anything was happening on my end at all. So
we only missed just the beginning. So please launch from the top.
no, I was just going to say that I completely agree with Sharmila about using about focusing on the relationship between our muscles and skeleton, because I really like what she said is you can cure all the diseases, but if your skeleton is not working, if your muscles are not working, you can't do anything, and this is so true. And on kind of like a related vein, since joining 10x Genomics, one focus of biology that I have become passionate about is immunology, because 10x Genomics has this product that can help us understand for any particular organism what does their receptors in their immune cells look like. So essentially telling us that, okay, for this person, this particular COVID vaccine would work best. So this is how my interest in immunology has evolved. And right now, we have such little databases on immunology. We actually do not have a big enough database that records all these different receptors, all these different genes that each of us have. Because what we have in terms of these receptors in our immune chains is each of us, we have our unique set of Lego pieces. And what our cells are able to do is cobble together these Lego pieces based on what virus is affecting us, what is attacking us. So it will put together pieces based on what we are facing. And each of us, we have unique Lego sets for this. Nobody has the same. So maybe I have some similarities with my parents and maybe in general with people from South Asia, but it will still be pretty different. My repertoire is going to look different from other repertoire. So there is a huge chance here for us to build these big databases for the genes that contribute to building these receptors, querying them, modeling them with AI, and then again, understanding like which antigen will bind best for which person, what kind of receptors should we be looking at. And this is also going to be huge for therapy, for therapies, for vaccine development. When COVID hit in 2020, right? 29, let's say end of 2019, 2020, by 2021, mid of 2021, we already had vaccines. Like that was already an accelerated timeline. I think with AI, we are going to see even more accelerated timelines, and that is going to be just fabulous. It's going to take so much of pain away from human life.
Nur, are you talking about TCR sequencing? Is that the term?
Exactly.
Exactly, yeah.
R&D.
You're absolutely right. And that's something that we really want to also incorporate, but each person has so many different TCR sequences, a great computational problem.
Exactly. Let's chat after.
Well, Eleanor,
Yeah.
you
So,
question.
obviously AI is right there with you, ready to take on the challenge. I wanted to ask the the three of you, and you've mentioned some of them, if you would share any other thoughts on beneficial deployments of AI, and then any sort of thoughts you have on your greatest concerns, regarding ethical and safe AI use. Anyone want to take that on? You're just jumping at it. I guess it's a two-part question.
Can you clarify the first part again?
Just thoughts on beneficial deployment of AI, some examples that you haven't already mentioned of where you're seeing it, sort of that the potential there, and then conversely reflecting on, what are your greatest concerns regarding sort of ethical and safe AI use.
So I know, so I can chime in a little bit. So there are a lot of people who research AI safety, and I think this is particularly an issue in making diagnoses and things like that with AI. You want it to have high certainty. But actually, I'm going to talk about something different. I was actually listening to NPR earlier today, and they were talking about how people are using vaccines much less. Maybe, the government or the current CDC is sending out mixed messages about vaccines. And sometimes they tell people to do their own research on vaccines. So a lot of people these days would just go ask an AI or something about vaccines. And now that can search up and just bring up anything. There could be one study with 10 people where, somebody had an adverse reaction or something like that. And that's not put into statistical context or anything. But currently, AI is not able to present the confidence in even that kind of information. And it's not able to accurately reflect the large body of research that's out there. And I think that's a real danger. If people are just asking AI scientific questions and the AI is not providing them with statistics, confidence levels, the sort of span of research papers. I think we all come across research papers where, the study has limitations, whatever it has. And I do think that people have to be cautious about using AI in this way because, also there are instances where the modern chatbots make up papers that don't even exist as citations. So these are real dangers.
Sharmila, you're unmuted. Is that because you want to jump in?
Yeah, I do want to jump in. I think the benefits, of course, we've talked about it, and there's nothing like making things more efficient or providing you with the answers. But sometimes the answers are wrong. They make it up. I don't know, maybe one of you should do a search on yourself. When I did a search on myself, it said I did my PhD from University of Michigan, Ann Arbor. It is unclear why it picked that. So it picked Ann Arbor for me. So there are things it picks which are correct, and there are things which it picks which seem extremely relevant. So consequently, I think AI is going to be using AI in any context, be it healthcare, be it in any context, is going to be like using any tool, is you have to know what you're using. It doesn't help to know, measure anything with a ruler if you don't know what the units are. It's going to be exactly that, only more complex because of the diversity of applications that you have using artificial intelligence. It is going to require appropriate training, appropriate knowledge, and exactly what data was your tool trained on. I think one of the biggest issues that we face, yes, it's beneficial, but if it's trained on data which is different, even either gender data or racially different data, geographically different data, it has a huge impact. Understanding some of these fundamentals is going to be really important in healthcare specifically because, again, the gender, race, all of these things affect your health overall and have an impact. So I think it's going to be, there are concerns, and of course, the data also changes. Sometimes the technology changes to monitor how your algorithm and model is changing when you have fabulous new data, great images or great photographs, so much better than ever before. Is the same model that picked out and improved your photographs from the 50s, is that same model going to work in the iPhone images of today? And the answer is probably not. So I think it'll probably have a detrimental effect. So understanding some of these aspects becomes critically important across the board. And that's why when you actually see a lot of models being used, for example, in healthcare, very clear indication of how were the cells prepared, how were the things prepared, how was it done, who were the people, what is their age, the gender. And Smita alluded to it, the statistics, the aspects of it have to be included in it. And I think as long as we have that awareness and we do this teaching, I think many of the concerns will be allayed over the in the future. But for now, those are just some considerations, I think. I wouldn't say they are my concerns, but more caveats which you have to look out for.
Yes, I agree with both Smita and Sharmila on this. And I am not going to belabor the point of the need for good data sets and genuine data sets that have been worked on carefully, but I do want to share this image that I today generated with ChatGPT. And see, I just asked it to create an image that shows and analyze single cell data represented on a UMAP for gene expression. And I have this. And this is not based on anything, any real data. And to the point of these two professors we have here today, like that is the danger. That is the danger of how easy it is to ask ChatGPT to just create a data matrix that is not based on reality. So we have to kind of make sure that that's not happening. One of the things that I think is good that I do see an initiative on this is, for example, when there are institutes right now, for example, the Chan Zuckerberg Initiative, CZI, and ARC Institute, who are working on building these atlases for cells with single cell data. And they are doing that in-house. They are only curating data that they're absolutely sure about has been worked on right and are being annotated properly. They are not taking data from all the papers that are being published out there. So they are putting some control on it. They're putting some guardrails on it, which is a good thing. I wonder if, I definitely don't want the process to slow down, but I think that if we could be careful about all the data that we are feeding into the training models, that is definitely one way. And then the other thing, which might lead to some more questions, is also when I started to work on bioinformatics and become a bioinformatician, one of the things that really worked in my favor was I also had the biology background. So I was kind of in this interdisciplinary area of I know biology and I can also use bioinformatic tools. And right now, one of my concerns is that a lot of the AI tools, because of the way that you build them, how coding heavy they are, etc., are being built by folks who might not be that knowledgeable about biology. And I think it would be really beneficial to have more career biologists involved in these kinds of initiatives and in even just the direct building of the tool, so that we can be sure that the inferences that we are drawing from these tools by just asking them questions with natural language and they're spitting out an answer at us, that is actually based in reality of the reality of biology and how biology works.
I guess I can maybe piggyback on that because it brought up a couple of things that I've been thinking about too. I totally agree with Noor. One thing is that when you're trained in the sciences, and I'm trained in some computational biology, but I don't have earlier degrees in biology. You're really looking for attribution. You're looking for cause and effect. So if you're predicting something, even you're classifying a T cell as a naive versus whatever, you can attribute it to something on the T cell surface, right? CD28, whatever. And this kind of attribution is really important. And we personally, whenever we redesign LLMs or any other kind of model for biology, we make sure that the internals of it are interpreted interpretable. So for example, in our immunogenicity model that I already showed, we have attention on which part of the molecule it's looking at and why it's saying it's inflammatory. So this kind of reasoning traces and things like that are very important. And I think those only get highlighted when people start to use AI, like people like me, computer scientists, we start to use and build AI for the sciences. When we don't do that, if the purpose of the chat GPT is just to chat with, lonely people or whatever it is, then you don't think about it. So these are real things. I think people should use AI for science more and look where its problems are, because that actually improves the AI. And I think interpretability, attributability are super, super important facets. And I'll one up Nur on on the lack of background of people that design these things. It's not even just like, I don't know, to me, it's like expecting them to know biology is a whole another ball game. I'm just hoping that they've even used bioinformatics tools before they have AI design some.
Agreed.
And so, and I won't mention anything specific, but I've seen, so there are AI agents out there, and you can say, okay, I have single cell data, how do I analyze it? And they just mimic. So they'll just mimic how some other paper did it and just spit it out to you. So that means every data set should be analyzed how some other paper did it, copied, right? So if a person who was actually a bioinformatician and had used these tools looked at that, they would be like, this is silly. It's telling me to analyze this differentiation data the same way as completely differentiated immune data or immune repertoire data. So I think that, yeah, so limited expressivity, mimicry, these are also problems. And I think when scientists use and develop these tools, these will also get better.
100%, yes. And it's not that each one of us can become, like, for example, I am not a computational biologist by training. I know bioinformatics, and that's kind of where it ends. But we need to collaborate more. We need to make sure that whenever we are drawing these, inferences, that's based in the biology for 100%. I think this calls for more collaboration and maybe even more training for all of us, so that we can work in these interdisciplinary intersections in a more robust fashion.
There is a question in the Q&A which is actually approaching a fairly general issue across all single cell genomics, imaging, across a lot of different things. It's saying there's a huge risk of bias and false training, and there are no guardrails for this. And I think contextualizing any output you get to the general population is extremely important. I think this is really very true. Today we have multiple models. There are vendors selling different algorithmic tools and various platforms, but exactly how the model was trained across in many disciplines, we really don't know, unless you have a really large, huge data set encompassing all examples of a particular thing, like all kinds of cells, or all kinds of genomic sequences, or all the images in the world. I think getting something which is going to be generalizable across and contextualizing that across a population does become extremely difficult. There are many, many new foundation models now which are being developed, which basically take huge data sets with multiple manifestations of disease and pathology and are attempting to do that. But even there, I think understanding and knowing exactly what the data was needs to be done. And today, there are no standards which have established that if you have an algorithm, what are the things you need to actually define before it is distributed. So I think the population needs to understand that. And that brings up, that really relates to the issue that NPR issue that Smita was referring to, is sometimes you get inadequate and non-generalizable information. So to respond to that question, I think, yes, the answer, your question or your statement is very true on the Q&A.
Thank you. Thank you so much. Now, racing to AI's defense and Chat GPT, Noor. Now, that image may not have addressed properly your area, but it was a fabulous paw print. I just want to thank you for sharing that. I'm a cat lover. You know, and it does bring up, you know, sometimes when I work with ChatGPT 5, I'm very aware that it's probably reflecting on its own obsolescence. You know, they will soon roll out a ChatGPT 6. I imagine there are several already versions ahead of that they're, you know, perfecting. I do wonder, and that's, you know, going back to a question I had before and one you all have touched on is, you know, what happens when AI hallucinates? You know, how do you see that in your research? You know, is it easy to flag? You know, it's, you know, we were talking about accelerating the pace and quality of biological research, and in terms of, you know, discoveries and improving, you know, the health of humans. Do you have any thoughts on that?
Yeah, you're talking about AI systems sort of hallucinating, as you called it, right? I mean, that's sort of a fancy word for your neural networks not answering correctly, and it probably is in relation to a generative model. It's generating some sample or sentence or something that's not realistic, that's actually not part of the space. So internally, what a generative model does is it has examples of real data or real entities, and it has to start from some kind of noise or some kind of initial setting and migrate over there. And it's probably going somewhere off what we would call the manifold of the real data. That's really kind of what's happening. There are ways of several ways of sort of flagging that. We, for example, whenever a neural network is operating, we look at its hidden layer representations. And if it's wandering around in space where there's no training data points, off in the wilderness, it's probably hallucinating. But also testing can really help. So you would test for generalizability in various domains. You can see how powerful the generalizability is. We often test on simulated models. So if we're testing, we have a neural network called Retini that infers gene regulatory interactions, but we have a set of real gene regulatory interactions that we've withheld or that we've modeled in a model system of gene regulation like Sergio, and we see if it reinfers those back. And so you can get a general confidence level from testing. You can just get a specific confidence level for certain types of inputs. And you can also, I'm a big believer in visualizing and interpreting the neural network as much as possible, the latent space and things like that, to see if it's wandering outside the manifold or in the manifold, and you know, these kinds of things.
I want to kind of piggyback off of that, Smita, with questions. So, for example, you have immunostruct, and you know, and you have tested it thoroughly, and you make sure that it behaves well. So, as a user of that tool, how can I benchmark this tool against others and say, okay, this is a tool that is proper and it has been tested, and you know, you have looked at where it is in the manifold. How can I, as a user, or users like me who just want to ask a question and get a quick answer, first validate? Because as we know, there are so many tools out there, not even AI, even if you look at just bioinformatic tools, there are so many out there, but some are definitely better than the other. So in this case, how are there any
I mean, so I mean, that's what we're here to do, right? When we develop a tool, we validate it, right? You can look at immunostruct, and it's been compared against every immunogenicity prediction tool that we saw out there that gives out data, even ones that hide their training data. And we tested it on data sets where we had immunogenicity predictions, the Cedar cancer data set, the IEDB immune cell data set. So the onus is on people developing those tools to validate it. But if you want to use it off the label, use it for something else, then you're redeveloping, and then you'd have to follow proper benchmarking practices. But we've actually created a whole system. You can look at a website called openproblems.bio, and we had a recent Nature Biotechnology paper about benchmarking in the single cell field. So we've created a repository of methods, so tasks, methods, benchmarks, and metrics. So there's many metrics that people could use. And so benchmarking is important. Some of that onus is on the people developing the tool. Don't use the tool if they didn't benchmark and validate it, right? Go ask them to do it.
Exactly.
Well, I love when you speak of the manifold. It makes me think of the matrix. But
Matrices, manifolds, all related.
Right? So we are nearing the end of our program, and I know it's so late for many who are joining us from the East Coast. We have had, many questions in the chat. I know each of you have sought to address them. I have a couple more open-ended questions that may respond to some. So really for the panel, are there any other things that you're seeing in terms of ways that scientists, are there any other ways you're envisioning the ways scientists will interact with AI in 5 years from now? Is it even possible to imagine? We'll be past the AI agents. They'll be quite advanced, but it's really on the human side. How do you see scientists, whether they're biological researchers or senior scientists like you, Nur, or Sharmila, people in your discipline, or if it can be sort of extrapolated to healthcare. Where do you see that? How do you see it changing, and how do you imagine it?
I think it'll become ubiquitous in healthcare in many instances, especially where it is a question of, for example, healthcare scheduling, assigning, exactly what the protocol should be. Some of the logic, it'll become ubiquitous once the workflow has been established for a given entity from that perspective. AI has become a part of us already. I mean, your iPhone does a lot of things based on your patterns and so on. So it's not new, it's emerging. There were some things we'll take for granted, but I think people will be using it. I don't think they will be replaced. They will develop new skill sets and the ability to use these tools more effectively. I assume that a single person sitting with real expertise can be impacting global healthcare, for example, because they can be sharing some of their findings through models. They can be running, you can remotely using AI and other remote connection tools, potentially be monitoring a lot of different things. So I think we might become more efficient. But again, as I said, this is all aspirational at this point.
I mean, I can say as a scientist, and a professor, I've already begun to use AI in some way. So if there's a new topic that I want to lecture on, so I often teach classes in deep learning, new trends in machine learning, and things like that. I'm also learning as I'm lecturing, and I can already ask AI to give me sort of a summary. I still see it can make mistakes and things like that. So that would probably become more comprehensive. It would probably give me a step-by-step breakdown of how some field was developed and how some question came to be, so making research process easier. I also wish it would do more to make sort of my life easier teacher, I get like hundreds of emails. Like it would be really great if it would sort them for me, auto respond to some of them. So I'm just really hoping that it makes the science, like the background research process and everything much easier, easier to go from idea to sort of communication of that idea or algorithm implementation, which some of my students still struggle with. So.
Yes, in my work currently, what we are one of the things that we are working on and is we can use the natural language processing of the AI to actually run our data sets through pipelines. So we can just input some things and say, hey, I have this data. This is my raw data. Can you please analyze this data with this particular software and then put the output files in this particular folder? So in that way, what I'm envisioning right now is for a scientist like me in the industry where we have maybe a few things that is dividing our focus. The AI is really going to be an assistant who is just like kind of always there for me, and I'm asking it to run analysis, go and compare some output files, as Smita mentioned, like summarize things for me. And in all these things, obviously, there will be a need for a human to validate within the next 5 years. Maybe things will improve way more after that, but at least within the 5 years, I see that constant validation is going to be required to make sure that what we are reporting on and working on remains true. But really, I think by 5 years, we are definitely going to have an assistant who is helping us with data analysis with the tools of our choice.
Fantastic. Thank you. I can't wait to check back in with
just
you
a little
in 5
amusing
years, but go
detail.
ahead.
Just a little detail is I think James Zhu, who's a professor at Stanford, has a paper on a whole lab of virtual people, like virtual grad students, a virtual professor and everything. So maybe we'll all be replaced by half virtual entities in a far future sci-fi view.
Love it. So do you have any other observations you'd like to share? And we had some questions in the chat, just wanting to be sure we get to all of them. But yes, Noor, I see your hand up. Are you just showing what it will be like when you're in a class of these, imaginary AI agents, and you'll be raising your hand and teaching them, no matter how smart they are?
Wow, that's something I have to wrap my mind around. But I'm raising my hand to ask a burning question, actually, I have for Smita, because I have had the pleasure of seeing her work at Yale and develop the different software and tools from her lab, which we have used to analyze single cell data. So one of my observations, and I've never spoken to Smita about it, but one of my observations has been that for like since a decade ago, she has been working on training data, neural models, neural networks, and and these were fantastic tools for single cell RNA-seq analysis. But I feel that most of biologists have instead gravitated towards using more easily digestible tools that were out there. I'm not going to name any names, but some more easily digestible tools that were out there for the single cell data analysis. But now with the tide of time, we have come to a point where actually the tools that Smita built now are way more relevant, and those are the powerful tools. So Smita, this is probably more of a career question. So how do you maintain your resilience in the face of, maybe seeing that, oh my, what I'm building is not getting as much, call outs as I would have hoped?
mean, it all kind of depends on your goal, right? So if you want to have your software just get a lot of downloads or something, you can spend all your time doing that, right? But our real goal is to push the frontiers of AI and computational biology. And so we're constantly trying to do that with it, and we're trying to communicate it in the best way possible. Every different model would be just to run like a repository and advertise it everywhere or something like that. And one of the most used models, which you use sort of yourself, UMAP, that's never been published, right? So that I would never do because I'm a scientist. I'm trying to push forward science. So I want everything peer-reviewed, published, disseminated, and then I want somebody else to build on top of it or something. And that's our real goal. And so we stay with our goal.
Excellent. Thank you for sharing.
I love this concept of panelists asking panelists. Does anyone else have a burning question for anyone else on the panel? How fun. Now I feel like we're back at Yale. No one. Sharmila, I was thinking since you were an early pioneer and innovator, you might want to keep Smita and Nur on their toes.
I think, the problem is that we are so much in agreement over time that, honestly, sometimes keeping people on their toes is tough in a situation where you all see the same thing in a slightly different discipline. I think that's one of the issues. And I think there have been a lot of questions and so on. So, I don't know. I don't have much more to ask or add other than it's been a wonderful experience, as somebody just mentioned on the chat out there.
Well, thank you. Well, we have 5 minutes. I think
Eleanor, there was also a question regarding the recording. I don't know if somebody got that about.
yes.
There will be a recording of this in case people want. And I think it has been a very energetic discussion, I have to say.
Wonderful, wonderful. Well, goodness, I so appreciate you all for joining us, the audience, and especially also our panelists taking your time, helping to develop this program. All of you, and I'm just going to give a special shout out to Nur. She's been really instrumental in helping to put this together. And an example of those special conversations, we were at a holiday party, she and I, when we met, and within about 5 minutes, I think I said to her, we should do a program with you. And, we, a few months later, put this together. So, I hope everyone has enjoyed it. And I look forward to reconnecting in future. And, this is also inspiring me to do another program on healthcare and AI. There's so much more to cover. So,
Another
so
holiday
again,
party is coming up.
yes, another one is coming. So, so thank you. Thank you so much. And I think we will. Does anyone else have anything to add? We're at 3 minutes, but maybe people will want to go to bed early, especially on the East Coast. Smita, where it's
That sounds good.
almost 10 o'clock.
Yes, yes.
Thank you. Thanks so much.
All right.
Thank you.
Take care. Have a wonderful night.
Thank
Thank
you,
you.
everyone.
Good night.
That's
Nice seeing
lovely.
Good night.
everyone.
Bye.
Thank you. Bye-bye.